This is where we left off. We're still inside the nucleus. We're still transcribing. We have initiated the creation of messenger RNA by pulling that DNA apart. Now, one of the things I want to clear up about this promoter signal. The promoter signal actually looks for that sequence, the TATA sequence, but there's also a transcription factors taking place. So the RNA polymerase recognizes and binds to the promoter, but the proteins called transcription factors mediate the binding and the initiation of transcription. The whole thing together, the RNA polymerase 2 bound to the promoter, is called the transcription initiation complex. And all of that happens in the TATA box, which they abbreviate as the TATA box or the TATA box. And that is the sum total of the initiation and the initiation spot with regard to this messenger RNA. And then, as I said, elongation takes place, and you have a nice trail. They call that downstream trail that is happening. And this particular graphic, you can see the initiation, and then at the bottom of the graphic you have this transcription initiation complex, which is the RNA polymerase 2 with these transcription factors that basically dictate the RNA polymerase 2 and dictate where it starts and how it attaches. And then what happens is the RNA polymerase, once it's attached, moves along the DNA as it unwinds, and it can do 10 to 20 bases at a time, and you can have multiple RNA polymerases on each gene, so that about 40 nucleotides per second in eukaryotic cells. So this can move along quite quickly, and so just remember that the nucleotides are added at the 3' end because of that chemical makeup with regard to the 3'5'. The mechanism of termination, so remember it's initiation, elongation, termination, is a little different between bacteria and eukaryotic cells. With bacterial cells, the RNA polymerase will stop at the end, the terminator at the end of the sequence, and can be translated without any kind of modification. So once it's created and the messenger RNA is created, the enzyme gets to the termination spot, and that's the end. With regard to eukaryotes, though, RNA polymerase will transcribe to an area, transcribes, it's called the polyidentation signal sequence, and that's a repeat sequence with regard to, it happens to be AAU-AAA, so adenine, adenine, uracil, adenine, adenine, adenine. That's the signal that it's time to terminate, it's time to cleave. And so at that signal, the RNA transcript is released, but it's released about 10 to 35 nucleotides past that. And then what happens is it cleaves, and then you have a tail that's attached. So you have this release sequence, and you can see this image. This doesn't come from your textbook, and it's the thing I want you to know. You don't need to necessarily know the polyidentylated messenger RNA precursor. Just understand that there is basically a termination signal, but that isn't the end of the story, because what happens is then that RNA that is still being associated with the DNA has to be destroyed. Otherwise, you're going to keep making a polypeptide, even though this one has been released. And we also need to then somehow finish off the ends, both of the 5 cap and then the 3 cap. And this this addition of the poly A polymerase tail, which we call the poly A tail, is going to be put on the 3' side end. So the messenger RNA then is released from the DNA. It's still in the nucleus. And we still are going to do something else to it before it actually leaves the nucleus and goes out into the cytoplasm. So what happens is before it's dispatched, there is going to be some parts that are going to be spliced out. We call those parts that are spliced out, they're called introns. And so you see a little bit of that image here, but you see it better here. So the pre-messenger RNA is modified in a particular way. That 5N receives a cap. That's going to be a specific cap that gets put on the 5N. You have actually a G molecule and 3 phosphates. Makes it a very nice unstable area with regard to that, but it tends to be the cap. I guess I'm assuming it's unstable. It's not something that I've read in your textbook. And then also you're going to put your poly A tail. The poly A tail is going to be attached to the 3' end. And then these modifications share several functions. They seem to help allow this messenger RNA to export, and they also protect the messenger RNA. So it's basically putting protective caps on it, and also maybe a little bit of something that will be attracted to the nuclear envelope and make its way through the nuclear envelope. They also these caps also will identify the 3' and the 5' ends so that the 5' end can attach to the ribosome. So they serve many functions in terms of these tails. Then there are in eukaryotic cells, for whatever infinite reason, it's not totally known, there are long stretches of nucleotides that are not coding regions. The non-coding regions are called introns there. There you see the word intron. And the coding regions are called exons, because they're the ones that express amino acids. And what happens is before this messenger RNA is released into the cytoplasm, these introns are spliced out. So it's kind of like, I've heard this, it's kind of like the exons are the television show you're watching, and the introns are what used to be, and maybe some of you don't even know what commercials are anymore, are the commercials. It's like splicing all the commercials out so you're just watching your show. And so there is a enzyme that splices them out, and it has a great name called a spliceosome. And what it does is it's an enzyme that can come in. The reason I'm looking at it, yeah, I'm pretty sure it's an enzyme. But anyway, it's a chemical that can splice those introns out and then allow the two exons to either rejoin, depending on what kind of splicing this is, or the exons can stand on their own if it happens to be alternative splicing. But the spliceosomes consist of several proteins and several small nuclear ribonucleoproteins. So it isn't quite an enzyme. It's a protein and ribonuclear proteins that it's kind of acting like an enzyme, but it really isn't one. And this particular chemical has the ability to recognize where the introns are and also do the splicing. And what you have here is a nice color-coded image of a spliceosome actually doing the job of cutting an intron out. And then once that intron is cut out, basically it isn't used for anything. It's degraded, I would assume. There is another chemical. They're called ribozymes. So look at that very closely, because these are not the ribosomes, which I've been saying over and over again. These are ribozymes. They are catalytic. RNA molecules that function like enzymes, and they can splice that RNA. So you do have, it's a ribozyme. Maybe it's a mixture, but anyway, it is. I don't want to confuse things, but just make sure when you see that word, you're not like me. And the first 10 times I saw it, I read ribosome. Ribozyme, it's what we've been talking about these ribosomes, but this is catalytic. It can act have the similar functions as enzymes and splice this messenger RNA and actually splice these introns out. Some intron sequences can regulate, so they're part of the game, and this is that part of that alternative splicing, where you have where the introns are inserted because the 3 exon pieces are domains of polypeptide sequences. And so what you have are these exons, instead of being reformed, they're all going to stand on their own. They're going to be separated. So what we have at the top is a long messenger RNA sequence that then is going to be cut into 3 sequences. Each of those sequences, the exon 1, 2, and 3, then is going to be go out into the cytoplasm and be used to actually code for 3 different polypeptides, the 3 different domains, and then those domains can get together in a certain 3D pattern to form a protein or not, but this is the nature of a protein. They have it's like the book says, a modular architecture that can consist of domains that get together. With this added variety or variation with regard to proteins, it's an infinite number of proteins that can be made by any given species. And so with this exon shuffling, the result is the evolution of new proteins. New proteins can be created all the time without any new DNA, without any new sequencing with regard to DNA. That's all happening inside the nucleus. So now what happens when that messenger RNA is ready to exit the nucleus into the cytoplasm, and now it meets up with a ribosome. So the genetic information flows from messenger RNA to protein through the process of translation. We're moved on to translation now. The cell translates messenger RNA with the help of another RNA molecule called transfer RNA, little t RNA. So we are introducing a new player, I guess, transfer RNA. And also we're introducing the ribosomes, and we are in a new setting. We're in the cytoplasm now. The transfer RNA brings the amino acid to the ribosome. The ribosome has clamped down. Remember, the ribosome is 2 units. You have a large unit and a small unit. And the messenger RNA is clamped into that ribosome, kind of like a vise. The transfer RNA has a amino acid attached to it. It goes and picks up an amino acid, I guess I might say, and brings the transfer RNA, comes to the ribosome with the amino acid, and it then acts as an anticodon. As remember, on that messenger RNA, you have groups of 3 nucleotide bases, which we call codons, and then the transfer RNA brings the complement to those codons, which we call an anticodon, which then corresponds to an amino acid, and brings that to the messenger RNA. And those amino acids are then attached to one another via bonds, and then you end up with a polypeptide chain. Translation is the complex process in terms of biochemistry. Now, that's not quite the definition of translation yet. A little bit more about transfer RNA on the next teaching time.